Hello everybody, welcome to NEET Simplified, your favorite NEET zone for simplified explanations of complicated concepts. So today I'm here again with another tough concept. So you're seeing the structure of gynoecium as such from the textbook. We have some terms like pistil, we have something called as gynoecium, we have something called as carpellary, multicarpellary, we have something called as syncarpous, and we also have apocarpous. Now this one single paragraph is almost showing 5 difficult terms to remember and also understand. So with this as the lead, let's understand each and every term and also understand about the development of Embryosac, which is a very difficult concept. So let's very clearly and easily simplify this. So let's go ahead to understand each of these terminologies. So here you are seeing a new diagram. So what you are seeing here is nothing but a gynoecium. So here you are seeing a gynoecium. Now we all know that we have got a seed in each flower, isn't it? We call that as thalamus. So here you are seeing a thalamus on which you have a gynoecium that is placed. Here also you have got thalamus and here also you have got a thalamus. Now this what you are seeing is a unit of gynoecium which we call as carpel. Right now on this, now yes, this is a gynoecium as you can see, and the unit is called as carpel. As you can see, there's only one carpel over here, and here this is also a gynoecium, but you can see 1, 2 and 3 units of gynoecium called as carpels, hence 3 carpels. So this becomes tricarpellary. Now coming to this one, this is a little important which you have to keep attention on. Now here also you are seeing gynoecium, you are seeing 3 carpels. Yes, this is tricarpellary as well. But what is the difference between 2 and 3, between the diagram 2 and 3? Here you are seeing on the seat, on the thalamus, there are 3 carpels, tricarpellary, which are not fused, isn't it? They are unfused, which means apocarpous. Apo in Greek means away. The carpels are away from each other, so it is apocarpous. Right now, if you look at this one, yes, this is also tricarpellary, 3 carpels, but you're seeing that they are all fused together, isn't it? So this becomes syncarpous. Syn is nothing but fusion in Greek, so we have fused. Whenever we have something fused, we use the term syn, isn't it? So this becomes syncarpous. So this is a syncarpous gynoecium with 3 carpels. Again, an apocarpous gynoecium with 3 carpels. Here also you're finding a single carpel in the gynoecium. Now coming to the term pistil. Right now we all know, or you might have seen at your home, a mortar and a pestle, isn't it? Now look at this pestle, which is definitely looking like what you are seeing on the screen, isn't it? Now you can just see here, when I put the picture and the diagram and the pestle together, you can see the resemblance, isn't it? And that is the reason why, because it is resembling a pestle, it is called as a pistil. Right now what you are seeing here is a pistil. Right here also you are seeing 1, 2 and 3 pistils, but here you are seeing 1, 2 and 3 carpels, as I told you, but they are fused together, and when you look at it you will see that it is something like a bulged pestle, isn't it? So this becomes a compound pistil. This becomes a compound pistil, right, with 3 carpels, and here you have got 3 pistils which are simple, right? 3 simple pistils we have got, and here also we have got 1 simple pistil, right? So that's about the term. What are the terms which we have uncovered here? Pistil, we understood syncarpous, apocarpus and carpel, right? Very quickly, what's this? Now this is our gynoecium with 1 carpel, the unit of gynoecium. It is 1 carpel present over here, which is a simple pistil. It is called as pistil because of its shape. And here the gynoecium has got what? 3 units. So 3 carpels resembling pestle. So 3 pistils. Here 3 carpels all are fused, looking like one bulged pestle. So one compound pistil with 3 carpels, right? So that's about carpels, pistils, syncarpous and apocarpous. Now let's go ahead and we're going to correlate this diagram with what we find in the flowers, right? What you're seeing here is the diagram of a flower. As you can see here, we have thalamus, just like here, right? Now we have the gynoecium here, isn't it? The carpel which we were talking about. This gynoecium with one carpel, just one unit, isn't it? So it is one carpel and it is simple pistil, right? Now let's understand here in the flower, we're also going to find something resembling pestle itself. We call that as pistil. Now this bottom bulged part, which is corresponding to this part, this we call it as ovary. This long tube-like structure which you are seeing here is called as style, and this bulged part corresponds to the stigma, which is basically the receptacle for the pollen grains, right? Now this is a simple pistil which you find in the family Fabaceae. Now coming to here, we have seen this diagram already, isn't it? Where you have seen that there were 3 carpels, tricarpellary, apocarpous, and we've also called that as 3 pistils in this gynoecium. Here also you are seeing 1, 2 and 3 of them, and this is gynoecium with 3 unfused which are away from each other, apocarpous condition, right? So all of these are 3 simple pistils. So we have multiple pistils in this one gynoecium. Now coming to this one, as we have seen the diagram already, now if you look at this one, you also can see 3 carpels which are fused. So syncarpous condition. So this is our gynoecium with 3 fused carpels called as compound pistil, which you find in Papaver and Hibiscus. Whereas in this one, as you can see multiple pistils, you can see that in the flowers like Michelia. So now I hope it is very clear about these terms now, right? Now let's understand or look at these structures in the real-time images, right? So let's understand how do they look when you look at them really. Now this is all diagrammatic representation so far. Now let's go and find out how they look like. Now in case of Fabaceae, as I've told you, it is a monocarpellary gynoecium as we have seen, isn't it? Now this is how it looks like. We have the ovary here, we have the style over here and the stigma. Now, if you want to know more about Fabaceae, we have a detailed video about this floral family. So the link is in the description. So please do understand that clearly as well, so that you will be perfect with the characteristics of Fabaceae as well. Right? Now, let us understand about the next one, the multicarpellary, as we have seen in the second diagram, Michelia, which is Apocarpous, isn't it? Now, here you are seeing multiple carpels, and as you can see, it might appear that all of them are fused, but they are just overlapping each other. You can see the ovary, the style and the stigma in each of these units, which are called as carpels, as we have discussed already. They are apocarpous. Now coming to syncarpous condition, here you are seeing multiple carpels in Papaver. As you can see here, there are multiple grooves which are representing nothing but the carpels, which are all fused. Hence it is a syncarpous pistil, which we will call this as a compound pistil, right? And even in Malvaceae, we have got here also a syncarpous gynoecium, which is usually pentacarpellary in case of hibiscus. When you take a cross section of the ovary, you will find 5 carpels. Yes, like this you are going to find pentacarpellary over here, and again syncarpous condition, hence a compound pistil. Now yes, as we have discussed, we should talk about something called as carpus, isn't it? Now where did this term come from? Now it's nothing but in Greek we call carpus is nothing but a fruit. Carp, in Greek it is fruit, right? Now what's going to happen to this ovary? You will see that ovary is going to be converted to fruit after first fertilization. Now, why is it called as ovary? Because it is bearing something called as ovules inside it, which in future would be the seeds after fertilization. So now we have carp, which means fruit, ovary turning into fruit. That is explaining carpus, syncarpous, apocarpus, or even the word carpel, isn't it? And now ovary having the ovules inside it. So the name is again justified, ovary having the ovules inside it. Now it might be that there can be only a single ovule in the ovary, or there can be multiple of them, like hundreds to thousands as well. Now, for example, if you take rice and wheat, these are actually the grains which you are eating are nothing but not seeds, they are fruits. Now, inside this fruit, you will find only one seed, which is nothing but this ovary had only one ovule, which is developed into the seed, right? Even in case of mango, we know the fruit is a single fruit with one large single ovule there, right? Whereas you might have seen in papaya, fruits like watermelon, here in orchards also, you might have seen there are multiple seeds present. That is indicating the ovary had multiple ovules present in there. Now, let's understand about, yes, we understood about the ovules, the ovaries, the terms are all justified now. Now, let's understand about the structure of the ovule, which is also called as megasporangium. Now, if you look at it, now for us to easily remember or understand, let's classify this part into, or this ovule, the entire structure into two parts, that is funicle and the body of the ovule. Funicle is nothing but the stalk, right, which is connected to the body of the ovule at a junction called as hilum, right? And this funicle is attached to the ovary wall through a tissue called as placenta. Now, for our better understanding, we are not considering the remaining part of the ovary. So, we are just removing all of that, and we are only focusing on the ovary wall part where the ovule is attached, right? So, this is placenta, through which you will see the funicle is attached and to the body of the ovary. Now, if you look at this, coming to the body of the ovule, you will see two protective layers protecting it. One, the outer one is outer integument, and the inner one is called as inner integument. And you have very important tissue which is called as nucellus, which we will understand about it in detail after some time. Right now, this outer and inner integuments are not continuous. They leave a gap which is called as micropyle. Now, here also you will see opposite to the micropyle on the bottom side, you will see that at this place which is called as chalaza, you will see that this outer and inner integuments are not differentiated. Rest of the place you can see that they are differentiated, but not here. And this junction is called as chalaza. Now, what you're seeing here is called as an orthotropous ovule, where you have to remember that micropyle, chalaza and funicle are in the same line. Now, such ovules are orthotropous ovules. Now, in some conditions, you will see this body of the ovule is bent 180 degrees downwards. Now, for that reason, we will call this as anatropous ovule, and in such case, you will find that micropyle and chalaza are in parallel lines, isn't it? They are coming parallel to the funicle. So such ovule is called as an anatropous ovule, right? Now that's about the structure of the ovule. Now we are going to the most important part of the video. So stay till the end to understand and to score definitely to crack 2 to 3 NEET questions directly. So stay tuned till the end. Now comes the most difficult part. I know it is very confusing. The development of Embryosac, which has got literally lot of steps in it, isn't it? So we are going to simplify that by doing one small comparative analysis with the pollen development, right? So we are studying development of embryo sac and pollen development together so that we can understand both and simplify very quickly. Now here, as you can see, I am showing the pollen development stages on the screen, but if you want a detailed video about it, the link is in the description. Do watch it so that you will be clear with that as well. Anthers, typically all the angiospermic anthers are containing 4 microsporangia, isn't it? So we call that as tetrasporangiate. So let us consider one microsporangia over there. So here what you are seeing is one microsporangium, right? And we have a tissue here which is called as a sporogenous tissue. And let's assume that there are some 100 cells in it. All of the 100 cells are going to be transformed into microspore mother cells or pollen mother cells, right? Now, coming to this part, here we have the development of embryosac. In this part, here we are showing the pollen development. Now, here in the ovule or in the megasporangium, we have got a tissue called as nucellus. And let's assume here also we have got 100 cells. Now, here you are seeing all of the 100 cells getting converted to microspore mother cells, isn't it? But here you will see only one cell getting converted to megaspore mother cell. Right? Now, what's happening here? We know all of them are doing work. Now, let us understand the process by just selecting or considering one microspore mother cell. Now, this is going to undergo a meiotic division, and we have a tetrad of microspores, right? Now, what is happening over here? Even this single megaspore mother cell is also undergoing meiosis and producing a tetrad, that is 4 megaspores, right? So, we have 4 megaspores over here. Now, what is happening here above? You will see that all of the microspores are going to be forming the pollen grains. But in this case, you will observe that the three of the 4 towards the micropyle end are going to be degenerated, and only one megaspore is going to be surviving, and this one is called as the functional megaspore. Right? Now, what's going to happen eventually? We're not showing the degenerated megaspores. You will observe that this single surviving megaspore is going to enlarge in size. So, this is where you will see the functional megaspore, which you have seen here, is enlarging in its size. And now such formation or such development of embryo sac is called as monosporic. Why do we call that so? Because there is only one megaspore, isn't it? One megaspore, which is eventually leading to the development of the embryo sac. Now let's understand what happens further in both of these cases. So here you can see these microspores undergo further divisions, isn't it? Each of them is going to form a pollen grain. So what are they going to do? Each of it is going to undergo mitotic divisions. Now this mitosis, you will observe that there is a karyokinesis but asymmetric cytokinesis. You will observe there is asymmetric cytokinesis in case of the mitosis happening in the microspores. So in this case, because of this asymmetric cytokinesis, we have a smaller generative cell and a larger vegetative cell. Now in this stage, we have a bicellular pollen stage, isn't it? You will see that more than 60% of angiosperms release the pollen in this stage where it is bicellular, larger vegetative cell and a smaller generative cell. And what happens in some of the plants is that the generative cell undergoes another mitotic division, forming 2 daughter cells, leading to the formation of tricellular pollen state, right? So in less than 40% of angiosperms, pollen grains are released in a tricellular pollen stage. Now coming to the embryo sac, what is happening? Here also there is mitosis happening, but there is no cytokinesis. You will observe only karyokinesis happening, leading to the formation of 2 daughter nuclei to each of these, right? And then we get a 4 nuclear embryo sac. Now further with one more karyokinesis, we get to the formation of 8 nuclear embryo sac. Because of the 4 nuclei undergoing karyokinesis, we have got 8 of them. Now, here, so far we have seen there is no cellularization that was happening, isn't it? Now, from this stage, when 8 nuclei are formed, then starts the cellularization. So, how does that happen? you can see at the top near the micropyle end, we have got two synergids. And there itself, we have another cell which is called as the egg cell. And towards the chalazal end, we have three antipodals. So far how many we have got? We have got two synergids, isn't it? 1 and 2. We have got one egg cell, that is third one. And we have got fourth, fifth and sixth antipodals over here, right? And in the center, we have got a central cell, which is this large cell in the part which is in between all of this, right? So this is called as the central cell, which is the seventh one. Now, after this, now we have got two polar nuclei over here in the central cell. Now, this towards the chalazal end, the polar nucleus towards the chalazal end moves just below the egg cell, right? So, with this, how many cells we have got? We have got 7 cells, but 8 nuclei. So, that's how we get our 7-celled 8-nuclear embryo sac. Right. Now, very quickly, let's understand how many divisions are happening in microspore mother cells and also in the megaspore mother cells. So, let's summarize it very quickly, which is very important in your exam point of view. So, here, if we see, we have got one microspore mother cell, isn't it? Now, this is undergoing one meiosis. As a result, we have got 4 microspores. Now, each of it is undergoing 2 divisions, isn't it? So, that becomes 8 mitotic divisions. That is 4 into 2. And because of this, eventually we get 4 pollen grains. Now, when you consider the next one, the megaspore mother cell, we have got one megaspore mother cell, which is undergoing one meiosis, right? And this is forming one megaspore. This undergoes 3 mitotic divisions. As we have seen so far, 3 mitotic divisions, after which we are with this stage of 7-celled 8-nuclear embryo sac. Right, I hope I'm clear, and if you understood it, do like the video and also share with your friends. All right, so what we have seen just now is a 7-celled 8-nuclear embryo sac, isn't it? Now, there are few other types of embryo sacs where you might find there are more antipodals, right? There are, so the cell number can vary. So, what we have just seen, the 7-celled 8-nuclear embryo sac is called as the polygonum type of embryo sac. And why this name polygonum? Very simple. It was first observed in a plant called Polygonum. So it got the name by the name of the plant. So we're going to understand the ploidy of this polygonum type of embryo sac. But what are we discussing here is the ploidy of this structure, which is very important and it is a little confusing. So let us simplify that as well. So as I told you, this ploidy is really confusing. So how can we really understand it through the diagram? Very simply, with a simple clue that the outer parts are all diploid and the inner parts are all haploid. Right? You can observe here that outer integument and inner integument, which are outside the embryo sac, these are diploid, and we have chalaza, funicle and placenta. Outside, isn't it? These are all diploid. Now coming to the haploid part, this part of the embryo sac which is all inside. Synergids, these are haploid, egg cell is haploid, polar nuclei are haploid, each nucleus of it is haploid, antipodals are also haploid. Now comes a little confusing part which you have to remember. The central cell is always represented as n plus n. Now when it is asked in examination, like we have options of n plus n, then definitely central cell is n plus n. If at all n plus n is not in the options, then we have to choose that central cell is diploid in stage. Right? Now, We have the haploid part over here. We understood that diploid part is all the outside. Now we also know that the part of the plant which is haploid is called gametophyte. That which is diploid is called as sporophyte. Now one simple thing to remember here is that in case of the ovule, we have got the gametophyte inside the sporophyte. So that's how we can remember the ploidy of this entire structure, right? And also, let me tell you that this part of synergids along with the egg cell, we will call this as egg apparatus. This is also very important for your examinations. And for our next video, let's very quickly understand about the structure of the synergids as well. So it has got a vacuole and the nucleus, and you can see some specialized finger-like structures, isn't it? So these are called as the filiform apparatus, which are going to direct the pollen tube. Right, so that's about the structure, and this is very important for you to remember. Only through the diagram you can understand it. So here, let's compare the terms in angiosperms and gymnosperms. In case of the angiosperm, we call the carpel, as we have discussed earlier, isn't it? We call that as carpel, a unit of gynoecium, right? And that is called as megasporophyll. So the equivalent term of carpel in gymnosperms is megasporophyll. Now coming to the ovule, which is Megasporangium with integuments together, which is the ovule, and it's the same way ovule in gymnosperms as well. Nucellus is also the same, and female gametophyte is called as Embryosac in angiosperms, whereas in gymnosperms it is not Embryosac. Instead, we call that as Archegonium, right? So with this, we are at the end of our discussion. I hope you vibed with us. If you liked it, share it with your friends, give us a like, and don't forget to subscribe to NEET Simplified. Keep up your efforts, continue that. Don't let any failure disappoint you. Stay consistent with your efforts, and I am here for you till your NEET success. Thank you so much for watching, and don't forget to hit the bell icon so that you are updated with our next video as soon as we upload it. Thank you. Bye bye.